So the general uh, rule here is as soon as Francesco starts talking, we ask you to mute yourself unless you have some questions. If you have some questions, then you should uh, just unmute yourself and ask them. I guess it's all right with you, Francesco, to ask,
Yes, of course, yes.
to interrupt you. So, uh, in otherwise, your talk should be between about 50 minutes and an hour, and then we will have some time for hopefully for general discussion and questions. But if there is something you want to ask while he's talking, just unmute yourself and talk. Okay, Francesco, I think you can get started now.
Okay, so thank you very much, Joel. It's really, as usual, it's a big pleasure and honor to speak in this series. So thank you for the invitation. I will present some work that we did over the past few years in collaboration with these people. As I was telling Joel before, we have a kind of a shared group between Rome and Paris, together with Martin White. This is our logo, and we have a GitHub where you can find codes, and we also have a little web page for this little joint group that you are welcome to check. So I will mostly present work that is published in these free references, and I try to prepare a modular talk. So there will be sections, and at the end of each section there will be a small take-home message. I hope this will help, and I will not probably be able to go through all of the sections, and this is okay. So please feel free to interrupt anytime if you have questions. So now, okay, so the first thing is the goal. So as you all know, in the field of in the study of proteins, the basic paradigm is the mapping from sequence to structure to function. So proteins are machines that perform functions, and physically they are chains of amino acids. So the idea is that you start from some chain of amino acids, which are represented as letters, and from the sequence, from this chain, then the protein will fold into a three-dimensional structure. So this is the sequence to structure part, and then this structure will then be present in the cell, in some part of the cell, and will perform some function. For example, some protein I will mention later is called beta-lactamase. Is a protein that is expressed by bacteria to degrade antibiotics. So when a bacteria is attacked by some antibiotic, it will use this protein to cut it and survive the antibiotic. So this is the function in that case. Now, as you know very well, the problem of mapping sequence into structure has been, let's say, essentially solved over the past years with the advent of deep learning. And there was a Nobel Prize this year for this reason. And well, one of the reasons why it was solved is that you have a lot of data. So now in public databases, we have about 10 to the 8 protein sequences of variety of proteins that perform all kind of functions. And we also have a lot of structures that are obtained experimentally, or of the order of 10 to the 5 structures. So you have a very good training set to train modern machines. And also the problem is somehow mathematically well defined, in the sense that the sequence is a sequence of letters. A structure is a collection of positions of atoms. So they are both very well-defined objects mathematically. And so you can use these machines. The problem with the last step, so when you want to understand function, there are two problems. One is that there is much less data available. So there are few experiments that are able to measure function reliably, reproducibly, and so on and so forth. So we have much less data than for structure. And also, the problem is that function is not as well defined as structure, because function can be many things. It can depend on the experiment, on the environmental condition, and so on and so forth. So, for example, if you have bacteria and you want to know if your protein is functional, you have to decide what is the level of antibiotics. Then you and so on and so forth. So it's a much more it's a much less defined concept. So what we try to do in our group, so this will be the long-term goal of our study, is to understand something about function from the sequence. So we try to skip structure and jump directly to function just for simplicity. Of course, we can try to integrate structural information into our models, and we do try, but it's difficult. So for the purpose of this talk, I will be mostly trying to skip directly from sequence to function. So the long-term goals are, as I said, learn something about the sequence to function relationship. Try to use this to design artificial protein sequences that have some desired functions. And I will be then more precise on all these things later. We would like to understand how nature does this through evolution, so how you can evolve functions that you want to have, and also how we can understand and possibly optimize protocols that do this in the lab. And these are so-called directed in vitro evolution. So when you try to evolve some protein in the lab to obtain some function that you want. So this is the take-home message for this first part. So the long-term goal of our group is to try to use data. So we use public data mostly, and develop models based on statistical physics to try to do something on these things.
But isn't structures a crucial part in the function? I mean,
yes, of course.
So,
okay,
I guess you'll explain how you can.
It's a very good point. So the idea is that structure is in principle encoded into sequence. So when we try to go directly from sequence to function, we use models, and these models learn something about the structure. Now, if you have explicit information about the structure, you can try to include it in the model. The problem is that for what I'm going to try to do, if you try to include structure, you slow down a lot the calculation. And so this is why we try to, we in the first step, we don't do that. But of course, we are trying to also include structural information at the later stage. The thing is that it's just computationally very heavy to include structure. So if you can skip it, it's much faster. And so you can hope that the sequence is already giving you some information on what is the structure.
Okay, thank you.
Okay, you're welcome. Okay, so this is the goal. Now, what kind of data can we use for this goal? So we have 4 main categories of data. So the first data we have is just natural data. So evolution over billions of years has produced a diversity of proteins that we see today. So the idea is that there was at some point some ancestor protein. So for example, these beta-lactamase proteins are quite old. So a long time ago, there was a protein that was doing this antibiotic resistance. And then it evolved over the time. And what we see today from public databases is a variety of proteins. So here each line is a protein that you can take from a different species, or maybe from different patients, and so on and so forth. So you have many proteins. You can align them, and all these proteins fold in basically in the same structure, and they perform the same function. And what is very impressive is that still you have a lot of diversity between these proteins. So if you look if you take two such proteins at random, they perform the same function. But if you look to the sequence, you see that about between 70% and 80% of the sequence is mutated. So you have different amino acids in the same position. So you have what is called a divergence, so a distance between sequences that is about 70–80%. And you have, depending on the protein family, you have something between 10⁻² and 10⁵ different sequences that you can find in public databases. So if you imagine that, so what we typically do, it's an assumption, we imagine that these sequences are generated from some probability distribution over the space of sequences. This kind of data give you a very good coverage of the sequence space. So it's like if you have, I will try maybe to use statistical physics language because this is my language. So basically, if you imagine like having an easy model in the paramagnetic phase, basically you can imagine that this sequence, you can think to this as many different configurations generated independently in equilibrium. So they will cover more or less all the available space at the temperature at which you are working. So you will cover basically all the entropy that you have in the model. So I will represent this very schematically in a sequence space. So this is the space of sequences. And maybe I can try this. This is the space of my sequences. And the vertical axis is the energy function. So low energy sequences are kind of more likely to happen. And what we assume is that nature has produced a set of very diverse sequences that are good. So they have low energy and they cover more or less a big part of the space. Still, there are few. There are few of them, but they are good examples that are scattered a bit everywhere in the space. So these are natural data that we can get from databases. The second class of data that we can use is what is called deep mutational scans. So these are experiments where people take one protein. For example, I mean, typical protein have a length of 100 sites. So you have one sequence of 100 sites. And then they do on each site all possible mutations. So you have 19 possible mutations per site. You have 100 sites, so it's like about 2,000 sequences that you have. So this is the number of sequences that you produce. They all have distance 1 because they are all one mutation away from a given reference. And then you can put them in the bacteria and you can measure the function. So these are done in the lab. So for example, if you want to do this on a beta-lactamase, you would take your beta-lactamase, you do all possible mutations, you put it in bacteria, you put antibiotics, and you check if the bacteria is surviving or not. And in this way, you can assign a fitness to each sequence. Okay, so these are very local data. Yes, please.
But this is a very restricted set, right? So because you are missing the pairwise interactions
exactly.
for deep. So and I know that you guys are working with pairwise interactions a lot. So this should be limited use for you. The deep
yes, well, it is still of use because we can check at least that we get the let's say the 1 point terms right. But also, as I will discuss maybe later, the effect of a mutation on a given site is still strongly coupled to the background. So for example, we have some experiments where people have done this for two very distant sequences. So you have one sequence here and you do all the possible mutations. Then you have another sequence very far away and you do all possible mutations. And in this way, you can check how the same mutation, what is the effect of the same mutation in two very different backgrounds. And in this way, you can measure, you can get an idea of the couplings that you have between different sites. I don't know if I answered to your question, but...
Well, what you're showing here is the same background, right? You're just changing one amino acid at a time.
Yes. So this is one experiment. In principle, you can do another experiment like this, and you can do it in another region
Oh,
of
fair
the
enough,
landscape.
fair enough.
That's the idea.
Yeah.
Also,
thank you.
now people start to do experiment where they start to be able to do all, for example, all pairwise. So this is really interesting, but for the moment, it's still very expensive to do so.
Thank you.
So, okay, this is the second set of data, and it's mostly very local data about the local landscape that you have. What I will be mostly concerned with in this talk is what is called in vitro evolution. So in vitro evolution, the idea is you start with a protein that you want to study. Again, let's suppose it's a beta-lactamase. So you put it in the bacteria and they are able to survive antibiotics. So you take this protein, you mutate it, and you can do this by doing PCR with errors. So you introduce a few mutations. You create a library that has maybe 2-3 mutations with respect to the initial one, each one in the library. You put these in bacteria, and in this way you put them in antibiotics, and those that have good mutations will survive. Those that have bad mutations will die. So you select those that survive, and you do it again. And you can do many cycles. So now people are able to do about 100 generations, and so they can create quite diverse libraries. So in these experiments that are like 2-3 years ago, but well, now 5 years ago, they were able to get from a given sequence, they were able to diversify up to 20% of the sequence. The good thing in these experiments is that then you can do a lot of sequencing, and so you can get very, very complete libraries with 10 to the 5, 10 to the 6 sequences. So these are experiments where you basically start from one configuration of your system and you explore the neighborhood of it. The last kind of data that we can use, I will not talk about it much in this talk, but we are trying to use them, are data where people take 2 natural proteins and they try to construct a path of single mutations that bring from one to the other. So you start from, for example, you start from this one, and then you make a sequence of mutations that will bring you to the other one. So these are some selected references that I like a lot, and we are trying to use this data to understand something about the shape of the landscape. So, okay, the take-home message for this part is that there are many data. So I think it's a very good moment for modeling. There are many data and many more to come. So these experiments are becoming cheaper and cheaper and easier and easier. They are still very difficult, but there are many more data that are going to appear in the next years. Still not enough to do AlphaFold, but enough to play with simpler models. Okay, so how do we try to model these data? We try to use generative modeling. So this is going to be the next part. So you all know what is generative modeling. Just to recapitulate, it's some model that you train on, for example, many images of human faces. You train this model. It's a kind of a black box. It will give you a probability distribution over images. And then you sample from the model and you produce new faces that do not exist in the training set. And of course, you can do the same with text. And this is what ChatGPT and similar machines do. So we try to do the same with proteins. So the idea is the following. We start from an alignment of natural proteins. So the ones that we get, for example, from natural evolution. So let's say we have 10 to the 5 sequences that correspond, that all fold in the same structure and they all do the same function. We align them. And so now we have this big matrix where each line is one configuration of the variables that represent the protein. And we consider them as I was saying before, as independent configurations. So from this, we use this as train set, and we try to learn a model. And then if we are successful, if we sample from the model should generate new sequences, and these sequences should be proteins that do not exist in nature, but still perform the same function as the ones that we used to train the model. So we are statistical physicists, so we try to use models that we can understand in statistical physics terms, and these are what are called Boltzmann machines. So these are probability distributions that are of the Boltzmann form, so exponential of some energy function. And the energy function can be expanded as usual as a sum of one body terms plus two body terms plus three body terms, and so on and so forth. To keep it simple and to be able to handle the number of parameters in the model, we only keep one body interactions and two body interactions. So why we do this? Because in this way, we can encode two kinds of properties of the system. One is what is called conservation, which means, for example, on a certain site, you really want to have a D, otherwise the protein is not functioning. So if you put a local field on that site that strongly favors D, then you can do that. But this is not enough. I will show why. So you also need to take into account what is called coevolution, which means that, for example, if you have a V on this site, you really want to have a T here. So this is a two-site correlation that you can describe introducing these couplings. And of course, you could add free body, four body, and so on, but then the number of parameters in the model will increase, and it's going to be much more difficult. So yes, there is a question.
Have you guys looked carefully into this three-body term and argued somehow that it's negligible, or is it just by the number of parameters because the model's too big?
So there are many studies. There are many studies that looked at free body terms. For example, this one in this paper from the group of Rama Ranganathan, they measured, they completely enumerated. They took two proteins that differ by 13 mutations, and they did all the 2 to the 13 mutations in between. And then from this, they get the complete landscape for this subset of possible states. And they find that free body terms are important. They are very sparse. So in principle, you should be able to identify a sparse set of free body interactions that are really important. Let's say for our purposes, two body interactions seem to be enough, but of course it would be very good to be able to add. The problem is that you have to find a procedure to only add the free body terms that you really need, because otherwise the number of parameters explodes and then you are not able to fit the model. So yeah, this is something that maybe... Okay.
Thank you.
So you're welcome. So I would like to highlight that because we are statistical physicists, we try to use these simple and interpretable for us models. And another reason why we do this is that there are not so many data. So if you try to use much more complex the models, then basically you cannot train them because there are not enough data. But also because these models are, I would say, more interpretable in the sense that, for example, we can relatively easily compute the entropy for these models. So if we want to know how many sequences the model can generate, we know how to do it for a Boltzmann distribution. There are other generative models for which it is much more difficult to do that. So this is, for example, an advantage of using this. They are energy efficient. We can train them on a single CPU in hours, and they are accessible to everyone. So our packages are all public. You can use them if you want. So it's kind of, I mean, we like that we can share the models. This is a bit different from what people are doing now in the industry. We hope that we can bring something. Maybe we will be totally outperformed by these big companies. I guess time will tell. So for the moment, what I can show you is some results from this paper in which I was not involved. So this is work of the Ramaran Ganathan group, together with Martin White, Rémi Monasson, Simone Cocco, and many other people. They did precisely what I was describing. So they started from an alignment of natural sequences. In this case, they were using the chorismate mutase enzyme. It is an enzyme that is used by bacteria to produce some amino acids. So you starve the bacteria of some amino acids, they have to produce them. If they can rely on this enzyme, then it works, otherwise they are dead. So you can measure the function in this way. And they did exactly this. So they took natural proteins, learned the model, a Boltzmann distribution, and then generated artificial sequences. So what you see here in the figures are the relative enrichment, which is how much the bacteria are able to grow with this protein. So each point is one protein. So this is the fitness of the protein, how much the bacteria can grow. And this is the energy that the model assigns to the protein. So high energy is bad, it shouldn't work. Low energy is good. Now, these are natural sequences. So the model is trained to give low energy to natural sequences. And so, as you see, the natural sequences are all at low energy, and about 30% of them work. They don't all work because these are natural proteins that work in some species. If you take it out of that species and you put it in another species, which is here E. coli, it is not always working, because maybe the E. coli is not able to express this protein, maybe there is some incompatibility. But let's say 30% work. These are the red ones, and the threshold is put because the distribution of fitness is kind of bimodal. If you make a histogram, and you can put a threshold and separate. So these are the designed sequences, the ones that are generated by the model. And you see that if you take the low energy part, it's very similar to what you get from natural ones. About 30% of the proteins work, and the rest doesn't work. And for this, you really need to have both the fields and the couplings. If you do it just with fields, it doesn't work. So you need the couplings. So this work really, I think it was really a great work. It validated this approach, and it showed that this approach works. These models are generative. Now, what we have been doing since then, in collaboration with Martin White, is to, well, first of all, we have efficient packages that you can use. So you can go to the GitHub and you can use them if you want. We devised some procedure to reduce as much as possible the number of parameters. So we try to remove the couplings one by one when they are not important. So in this way, we can get to models that have maybe 10% of the parameters of the full dense model and still function well. We are testing them in collaboration with the group of Ramaran Ganathan. So if you're interested, there is a reference here. And in this study here, we tested this on a RNA, not a protein, but it's very similar. And we showed that the model works, and it is not generically outperformed by other architectures like generative adversarial networks or variation autoencoders, deeper networks. I mean, basically, these models have a success rate that is the best. And the other models can do the same, but they cannot do much better. So this is motivation to stick to simpler Boltzmann machines. So, okay, so the take-home message from this part is that simple generative models can design artificial proteins by equilibrium sampling. So you learn the model on natural data, and then you sample in equilibrium from the model, and you reproduce the same properties of the natural sequences. And now the question is, can we use these models to get some insight on the dynamics? So up to this point, we have just done equilibrium sampling, and now we would like to do some dynamics. So this is going to be the next part, dynamics. So again, we are a statistical physicist. If you have a probability distribution, the first thing you would like to do is to do some Monte Carlo to sample from it. So then, I mean, the natural idea that comes to one's mind is, can we try to use Monte Carlo dynamics to mimic an actual evolutionary process? We were not the first to have this idea. So in this is, to my knowledge, the first paper where this idea was proposed from the group of Farouk Morcos. What we did was to try to use this to describe some experiments. And in order to describe the experiments, we realized that you cannot do the Monte Carlo on the amino acids, because in the experiments, the mutations are done, and also in nature, the mutations are done on the DNA. So you have to do the evolutionary process, the mutations at the level of the nucleotides. So we devised, in this paper and in this one, we devised a procedure that you can use to do mutations on the codons, on the nucleotides, on the DNA, and then accept with the energy function that we learned on amino acids, and still satisfy detailed bounds in such a way that at long times you will be sampling in equilibrium from the Boltzmann distribution that we learned. So this is a bit technical. It can be done, and you can find it in the papers. So now then, what is the pipeline? So we would like to use this to describe experiments that are done in the lab. The ones that I was describing before, where you introduce mutations, select the bacteria that survive, do other mutations, and you go on like this. So what is the pipeline that we use? So you start from some sequence that is the one that from which you also start the experiment. We do homology search, so we look for other sequences in the public databases that have the same function. So we construct a family of natural sequences, and from the family we learn the generative model that we, as I was saying before. Once we have the model, we go back to the wild type, and from the wild type we do Monte Carlo. We do Monte Carlo steps, and we, for simplicity, up to this point, we do Monte Carlo chains in parallel. So we do many chains that start from the same initial configuration. We do mutation selection, and when we produce a library, and we can compare this library to the one that is obtained in the experiment. Now, this can, I mean, it is not trivial that this will work, because of many things. I mean, first of all, we learn the model on natural data. So this is different from, in principle, the natural evolution happens in different conditions from the evolution that you have in the lab. And we neglect phylogeny, so we do independent Monte Carlo chains. I mean, now we are working on including phylogeny, but I will not discuss it today. So once again, this is the experiment. You start from the wild type, you introduce mutations, you put the bacterial antibiotics, you select, and you do it again and again. And so in order to describe the experiment, we have two parameters that we need to calibrate. One parameter is the temperature. So when we do the sampling, we can introduce a temperature parameter in order to tune the amount of selection that we impose. Precisely because in the lab, the selection is not necessarily the same than in nature. So how do we do that? We have the experimental library. These are the blue data here. We assign to each sequence in the experimental library an energy using the model. And then we fix the temperature in such a way that the average energy of the library we generate from the Monte Carlo is the same as the one that we get from data. So as usual in statistical physics, you fix the temperature in order to fix the energy. But you see that we also fix, I mean, once we calibrate one number, which is the temperature, we can reproduce the full distribution of the energies. And then the other parameter we have to calibrate is how many steps of Monte Carlo we need to do. But this is easy. From the experiment, we know how many mutations we have in the experimental library after a certain number of generations. So these are the blue data. We calibrate the number of steps in such a way that the average number of mutations is the same. And once again, we can reproduce the full distribution. So in this way, we can calibrate two parameters. And once we did that, we can compare the experimental library to the library we generate from the model. Okay, is that clear? Sorry, my audio is not, I mean, there is no noise anymore, so I'm not sure if you are still there.
Maybe
Yeah,
I
still
can,
there.
maybe I can
You are still there.
Yeah.
Yes.
Maybe I can ask a question. So it's not clear to me what the role of detailed balance is here, because the actual mutations in the in vitro assay probably don't satisfy detailed balance. There's no reason why they should, right? So you have to impose detailed balance because you somehow want to stay within your generative model.
Yes, exactly. So in the first study, we actually use the dynamics that did not satisfy detailed balance. So, and it still works to describe the experiment on short scales. So the reason why we introduced detailed balance in the second study is that we also wanted, as you said, to stay in the generative model. So we want the Monte Carlo chains at long times. We want them to converge to something that has the same statistical properties of natural sequences. Maybe it's a wrong assumption, but I will maybe come back to this a bit later. But yeah, this is the idea. We wanted to have something that on short times can reproduce the experiment done on short times, and on long times can reproduce what we observe in nature. So we try to interpolate between the two. Maybe it's not the right thing to do, but we can discuss.
Okay, thank you.
Okay. So what I was saying is, okay, now we have these two libraries, the one produced in the experiment and the one that we get from the simulation. What we did is to take each site. So this is a protein that has 200 sites. You see here two experiments. There is one experiment done in Italy on this TEM1 protein, and another experiment done in the US on these PSC1 proteins. These are two beta-lactamases. They have the same length here because they can be aligned, and they are very different. So the two starting points of the two experiments, these two proteins have 70% mutations, are 70% mutations away. The two experiments did more or less the same thing. And so we have two libraries, and we can do twice the numerical simulation. And then on each site, we can compute an entropy. Basically, we look to the frequencies of amino acids on that site. So we take site 100, and we count how many times there is an A, how many times there is a C, and so on. And from these frequencies, we can compute an entropy. So now we have, for each experiment and for each protein and for each site, we have one number, and this number gives you the mutability of that site. So the sites that have very low entropy are very conserved. They don't mutate in the experiment. And the sites that have high entropy are the ones that mutate more in the experiment. And what you see is in purple, you see the experimental data, and in orange, you see the data from the model. So you see that once you calibrate three parameters, which are basically, okay, the temperature, the number of steps in the Monte Carlo, and the number of sequences, so you can decide how many chains you do. With these three parameters, you can fit about 400 numbers. So two experiments, one number per site. And you see that the model works quite well to describe the outcome of the experiment. So we were happy of this result, because at least we can trust a bit the model. Okay, so what next? You were asking about the importance of decoupling. So this plot is supposed to show the importance of decoupling. So let me try to explain what is this plot. So let's forget about the model for a moment. So this is just experimental data. So we have two libraries. Each library was built by mutating one natural protein. So these are the two natural proteins, and then you get a library of mutants. And now you can compute on each side this entropy. So this is a scatter plot of the entropy in one experiment versus the entropy in the other experiment. Now, if the mutations were happening independently on each side, so if you had no coupling, then the entropy should be the same in the two proteins, because each side would mutate independently of the rest of the protein. So this is, in many cases, this is true. So the two numbers are on the diagonal, but there are also quite a lot of sites that have very different entropies. So here entropy is here we normalized it to 1. I mean, so it's entropy divided by the logarithm of the number of amino acids. And you see that, for example, this site in one experiment is very conserved. It doesn't mutate much, but in the other experiment, it mutates a lot. So this is due to the fact that the rest of the protein is very different. And so the couplings between sites make this possible. Okay, so this is what in biology is called epistasis. And it means that the effect of a mutation at a given site depends on the rest of the protein.
Francesco, can I ask one more question?
Sure.
The experiment, so you say genetic drift, but what are the selection conditions? So are these beta-lactamases both well adapted to the antibiotic that you're exposing them to?
Yes, so the initial proteins are, I mean, they belong to these superbugs class of proteins they are sequenced from patients that have been hospitalized for very severe infections. So they are very adapted to the antibiotics. So the experiment is done at a level of antibiotic that is comparable to the level that these proteins can survive. So it's neutral drift. Basically, the proteins that are produced in the experiment have a comparable fitness to the one that was used initially. And this is why when we model we use a temperature. I mean, when we fit the temperature, we find that the temperature is very close to 1. That was the training temperature. So basically, you are sampling. I mean, you are doing the dynamics at the same selection that nature has used to produce these initial states. There is an experiment. This one. Sorry, this one. This is a new experiment. I mean, it's 2 years ago, but it's still not well analyzed. Where they did first high selection, so very high amount of antibiotics, and then they relaxed it, and they studied the response to the evolved proteins to the change in temperature, basically. So it's a response experiment where you can do, and this is very interesting, and we are trying to analyze the data using this model, doing some temperature cycling, basically.
Yeah.
So for the moment, we don't have still results, but we are working on it.
Yeah, thank you.
Okay, and so, okay, just to conclude this part, this slide is to mention that this is for example, this plot shows how two Monte Carlo chains are diverged as a function of Monte Carlo steps. So you see that initially they are not very diverged, then they start diversifying, and then they slowly reach the asymptotic value, which is the one of the natural sequences. So the experiments are in this range. The experiments can reach about 20% diversity. So you do, so we model it with the Monte Carlo in this part, and nature is kind of here. And what we would like to do next is to try to use our model to interpolate between the two, precisely as you were asking before. So the take-home message of this part is that we can do Monte Carlo dynamics on these generative models that we learned from natural data, and we can use it to describe in vitro evolution in the lab, and it seems to work quite well. So this is short time. Now, I can take maybe 5 more minutes to show you this last part about epistasis, so about the importance of the couplings. So as I was saying before, in the experiment, you see that there is epistasis. So different backgrounds give you different mutability on the same site. How can we quantify this effect? So we introduce 2 entropies. The first entropy that we introduce is the one I was telling you before. So we take the natural alignment, we look to one site, we look to the frequencies of the amino acids on that site, and we compute an entropy. And this is easy. So this is just from the data. This is obtained from the data.
I mean, how do you
sorry.
To compute the entropy, you look at each site?
Yes, we do it site by site. So this is just so we want to identify sites that are strongly coupled to the other sites. So the first thing we do is, I mean, suppose your model was, if your model was a model of independent spins without couplings, then each site is independent. You can compute the entropy site by site, and the total entropy is just the sum of these local entropies. So we start by computing the local entropies site by site from the natural data. And I mean, we can also do it from the generated data. It's the same because the model is generative.
Okay.
Okay. The second quantity is what we call context-dependent entropy. So this we call context-independent entropy because it's just one site forgetting about the rest. This we call it context-dependent entropy because we take one site, we fix a background, so we take one protein, we fix all the sites except one, and we look, I don't know if I have the formula, sorry, no, I don't have it. Anyway, we look to the probability that the model assigns to each amino acid on this site, and we compute the entropy of this probability. So it's like if you do a single Gibbs Monte Carlo step, you freeze everyone but one variable. You can look at the probability of putting variables on this site, and you compute the entropy of this. If the entropy is very low, it means that the model is telling you, given this background, this site cannot mutate. If the entropy is high, then the model is telling you that this site is free to mutate. So we can compare these 2 quantities, okay? If they are both small, it means that there is a site that is conserved in the natural alignment. So for example, you always have a P, and also whatever background you put in the rest of the sequence, you can only put a P. So this is a site that is trivially conserved. There is a strong local field that is favoring a P, and nothing else can be there, okay? Then you can have sites that have both quantities very high. This means that they are very variable when you look to the, say, equilibrium samples, but also if you freeze everyone but one, you see more or less the same variability. So these are very variable sites, but they are still very, but they are basically decoupled from the rest. It's like you have an Ising model, and there is one spin that has 0 couplings with all the others. The interesting ones are these ones that have high context-independent entropy, so they can mutate a lot if everyone is allowed to mutate. But if you freeze all the rest of the protein, then they are constrained. So these have very low context-dependent entropy and very high context-independent entropy. So these are sites that are strongly coupled to the rest of the protein in such a way that they can only mutate if you allow the other sites to mutate. So up to this point, this was a scheme, but these are actual data. So we can take the model, we can take the PSC background that was the one used in the experiment in the US on the beta-lactamase, and we can make a scatter plot. So each point is a site. And you see that you have a bunch of conserved sites. These are really, they don't mutate. Whatever you do, they will never mutate. You have a bunch of variable sites that are very floppy. They can mutate in any case. I mean, even if you freeze all the rest, they will still mutate. And then there are a bunch of epistatic sites that can only mutate if everyone can mutate, okay? So the last thing we did, the last thing I want to show, is this plot here on the bottom of the slide. So what I show here is the entropy of each site during the Monte Carlo simulation. So I start from the PSC wild type, the beta-lactamase protein. I evolve the library, and I look to the entropy of each site. So for the sites that are conserved, we don't see any mutations. So you see, at all times, these sites have very low entropy. The sites that are variable, these are very floppy, and they're very decoupled from the rest. So what happens is that very quickly, they go to the equilibrium mutability, they very quickly mutate, and they reach their natural diversity, and they stay there. What is interesting is what happens to the epistatic sites. So these sites start mutating a little bit at the beginning, but still, because the protein has not mutated much, they cannot mutate much. So the entropy stays low, and it takes a really long time to reach the asymptotic value. Not only it's a long time, but it's also very heterogeneous. You can see that you have about 1 decade in time scales to reach, I don't know, half of their mutability. So these sites are very heterogeneous. There are sites that are more strongly coupled and sites that are less strongly coupled. Now, the region that is access to experiments today is more or less this one. So on this region, you see that the epistatic sites are basically conserved. You cannot do in these experiments enough mutations to start seeing the couplings. So you can see the epistasis because you can see that if you start from 2 different backgrounds, you have different results. But if you start from one background and you look how it evolves, the sites that are constrained remain constrained. In natural evolution, you can see something, but you don't have the short time. So what we hope is that in the next years, we will have some data in this region, because this is what really the model is telling us is the really non-trivial region where you have these interactions that really play a very important role. And there are experiments now that start to be able to do like 500 generations, maybe 1,000 generations. And so we hope that we can reach more or less this region here, and we hope that we will be able to check if the model makes sense in this intermediate range. So this is what we are trying to do now. And so the take-home message for this part is that epistasis, so the amount of interactions that you have, is very heterogeneous across sites. So in a given protein, you have a very different behavior if you look at different parts of the protein. And in order to see the correlations, you have to look to intermediate time scales that are still a bit beyond what we can reach in experiments. And well, this is more or less where we stand now. The last part that I will skip is just measuring heterogeneity across sequences. So we are looking to how different initial states give you different behavior. And what we find is that also it's very heterogeneous also across different initial states. So if you do 2 experiments with 2 different initial proteins that belong to the same family, you can have very different outcomes because the space of sequences is very heterogeneous. So we are working on this, and we hope that we can give some insight on how different initial states behave. I will close with this little advertisement for this book that we wrote with Simona Cocco and Remy Monasson, where we used it for a course at the master level, and it contains a bit of introduction to these ideas about some machines, analysis of biological data using statistical physics techniques. So if you have feedback on this book, we are happy, and if you want to use it, we are also happy to provide additional material and so on and so forth. And I have a few copies, so if you ever come to Rome, I can give you a copy. And with this, thank you very much for your attention. Thanks again, Joel, for the invitation. I hope you enjoyed the seminar.
Thank you very much.
Thank you.
Francesco, let us unmute ourselves and give Francesco a hand for a very, very interesting talk. Why don't you stop sharing the screen so we can open the thing for questions or comments? Are there any questions or comments?
Yeah, let me start. So this is very interesting. I'm happy that I could see this. I was wondering about the last part about epistasis. So you're basically quantifying epistasis through the entropy, right? But since you're doing Monte Carlo simulations, so you actually have a trajectory, right? So you could also look at let's say selection coefficient. I mean, the other thing I noticed is that you haven't used the word fitness throughout your talk, right? But in the end, of course, the log of your probability is something like a fitness. So if you sort of, at least I would imagine, if you have a state that is being accepted, then this means there is a kind of positive selection coefficient for it, right? So you could look at correlations between selection coefficients and for example, distinguish positive and negative epistasis and things like that. Is that right?
Yes, exactly. I'm sorry, I didn't prepare a slide on this, but I can try to find a figure. But okay, basically we try to do something like that. The reason why we mostly work on the entropy is that we try to average a bit the noise, because when you look at the level of individual mutations, the problem is this. Let's say it in this way. If you think in statistical physics terms, the couplings that you have in the model are look more like what you would have in a Sherrington-Kirkpatrick model. So you have many weak couplings that together contribute to a strong epistatic effect. So if you try to isolate one single coupling, you typically find that it is quite weak. There are a few stronger couplings that are very sparse and that typically correspond to like physical contacts between amino acids, strong coevolutionary signals that come from typical structure. Most of the epistatic signal comes from this distributed coupling. So if you look to individual, for example, we looked along trajectories, we look to delta E, and we look to delta E to try to find epistasis. And there is signal. I can look for a figure, but you can also find it in the paper. There is a signal where we show the delta E as a function of time, and we see a signal that looks a bit like what, for example, we compare it with what Joe Thornton has found so there is a very beautiful set of experiments by the group of Joe Thornton where they reconstructed an evolutionary trajectory of some protein, the DNA binding domain. So they have a series of proteins along an evolutionary trajectory, and then for each of them, they have 8 proteins. They did a deep mutational scan. So they know the effect of each mutation in 8 different times, let's say, along the trajectory. So they can compare the delta fitness along this, and they find the signal, and we find the comparable signal, but it's a very weak signal. So basically, in order to get rid of the noise, we use the entropy just to average over the amino acids and just look at sites. But yes, you are right, we can do that. It's just very noisy.
And can you also see in that context this effect of entrenchment that people talk about, so that a certain a mutation happens and then it sort of gets entrenched as you go along and it becomes more difficult to
yes, absolutely. Let me try to find sorry, okay, I have it. Okay, I will share again my screen. Sorry for a second. So this is the paper. Sorry, so this is the paper, the second paper I was mentioning. So we have, oops, no, wait, it went, we had some, no, okay, it's here. So this is entrenchment. So this is the delta E. So you do a Monte Carlo simulation, and then you try to, at some point, after some time, you take some mutation that happened at time 0, and you try to reverse it after a time t. And this is the delta E as a function of the time between when the mutation occurred and when you try to reverse it. So the idea is that when the mutation occurs, it's typically close to neutral. And then after some time, it typically becomes deleterious. So you have an effect. But you see that this is 10 to the 3 is the time scale of the typical experiments that we have. And you see that the effect, this is the average, and this is the variance. And you see that there is an effect, but it's quite noisy.
Okay, very nice.
Mm-hmm. Yeah. Are there any other questions or comments? Well, I guess in all of these things, you don't use any quantum mechanical considerations. It's purely classical type of formula.
No, I mean, I think in the structure, I mean, people, when they look to the structure, they use potentials that derive from quantum mechanics. But for us, the structure is not, I mean, we don't use it explicitly. It is contained in the couplings. So as I was saying before, for example, there are a few strong couplings that are typically related to amino acids that are close by. So if you mutate one, you have to mutate the other. And then there is an interaction energy that contains quantum effects, of course, because, but we don't model it explicitly. We try to include a bit of structure into our models, and to do this, you have to do the following thing. You have to construct a machine, you need some model that gives you, given a sequence, gives you a structural score. So there are models that tell you, for example, based on structure, if you have a protein and you want to make a mutation, would it fit? For example, does the new amino acid fit in the cavity that you have? So these are effective models, and we could include them when we do mutations. We could include a score coming from the structure. The problem is that these are heavy. I mean, these models maybe take a few, I don't know. I mean, one step Monte Carlo for us is 0. I mean, the computer is like, is flipping one spin and computing L couplings. If you include the score, you typically slow down the simulation by a factor, I think, 10 to the 4, 10 to the 5. It's really much, much slower. So unfortunately, this is what I was saying at the beginning. Putting structure into the pipeline explicitly is not very easy, but we are talking to people who are expert in structure to try to find efficient ways to include this. So in an effective way, we try to put it inside, but.
Thank you. Are there any other questions or comments? Well, if not, let's again thank Francesco for a very, very nice, interesting talk. And thank you all. See you next week. Bye-bye.